Hello, welcome to Coursera, welcome to Caltech, California Institute of Technology, welcome to Drugs and the Brain. My name is Henry Lester, and I serve as the instructor for this 7-week course, along with a dedicated group of assistants and specialists here at Caltech. The first 2 mini lectures of this week 1 are overviews and organizational previews of the course, and in this lecture I will briefly forecast weeks 1 through 3, which give you the basic science of drugs and the brain. First of all, we start with the question, what's a drug? And we will discuss 4 typical drugs that serve as models for several aspects of drugs in the brain. Nicotine, the addictive drug in tobacco, lidocaine, a local anesthetic, morphine, an analgesic and an abused drug, and botulinum toxin. Note that we don't use trademarks. They vary by the country, and by the preparation, by the salt, and by the intended use. Note also that we give references to a database called PubChem, which is maintained by the U.S. National Institute of Health, free of course, funded by the U.S. taxpayer. We also give references to the protein data bank when we discuss protein structures, and these files are curated by an international organization. The fellow students and you will be organizing several web forums which tell you how to access these databases, how to display the molecules on your particular device or your particular computer and your particular operating system. In general, these viewers either are integrated into web browsers or cost very little as an add-on, as an app. We then move on to the question, what is the brain? We'll introduce the brain not so much in detail as I would like, but we will discuss the brain, which is part of the central nervous system, and so is the spinal cord. We won't emphasize the spinal cord, and we'll talk about such words as rostral and caudal and dorsal and ventral. Next, we bore down into the very most important organelle, the very most important contact between 2 nerve cells, 2 neurons, the synapse. An adult human brain contains nearly 100 billion neurons, roughly 10 to the 11th of them, and each of these might receive or make 10 to the 3rd synapses apiece, 1,000 synapses. So we multiply 10 to the 11th times 10 to the 3rd, and we conclude that a brain has 10 to the 14th synapses. Now, most of these synapses actually form during the first 2 years of life, during gestation and during an infant's first year. And so we can divide 10 to the 14th synapses by 10 to the 8th seconds in those 2 years, and we come to the conclusion that a fetus or an infant is making roughly 1 million synapses per second. No wonder they are so tired and need so much help. Then we'll move on to the topic of drug receptors. The example that we will return to several times in this course is the nicotine receptor, formerly called the nicotinic acetylcholine receptor family. Here again is a protein database link that gives you the file which can be displayed this way. We'll talk about the various parts of the receptor protein, and if we now focus on one particular part of the receptor protein, an interface between 2 of the subunits, we actually see the area where nicotine itself binds. We'll talk about the kinds of non-covalent, reversible bonds that are made by nicotine. We'll show you how to display graphics like this and how to do some measurements. So then we are armed with some of the basic concepts, and we talk about the fact that drugs activate ion channels. A good example is nicotine activating the nicotinic receptor. How this activation comes about is not exactly sure. Here is one example, giving as much detail as we know at the moment about the parts of the receptor protein that communicate with each other. Other parts that look like alpha helices. We'll describe alpha helices a little bit more. And so that is just about the most detail that we can give for activation of an ion channel by a drug. Another important part of drugs in the brain is that drugs block ion channels. There are many examples in drugs in the brain: antiarrhythmics, anti-epilepsy drugs, local anesthetics, all act almost as though they were plugs in a drain. The plug is the drug, the drain is the ion channel. When the plug is out of the drain, the ion channel conducts ions, current as usual. These are stylized records of single channel traces. We'll show a few more of those traces from individual ion channels. But when the plug is in the drain, the plug is in the ion channel, no current can flow. And we'll show how these processes block signal transduction, desynchronize signal transduction in the brain. We'll then move on to the very important topic of drugs that act on G protein pathways. Here is a molecular structure, again with a PDB file, of a beta adrenergic receptor, a G protein coupled receptor, shown schematically here. The structure is complexed with its G protein, shown schematically here. And ultimately, not shown in the structure here, is an effector, an enzyme or an ion channel that actually does the business in the signal transduction within or on the surface of cells. There are several varieties of G protein coupled receptors and of effectors. We'll talk about most of them. Downstream from the G protein coupled receptor and from its effector is some interesting intracellular biochemistry. To measure that intracellular biochemistry, we can do assays on broken cells, or we can do assays, experiments on live cells that are labeled with fluorescent versions of the intracellular messengers, and we'll discuss experiments of this sort during the first 3 weeks of the course as well. Another important aspect of drugs in the brain is the actions of some drugs on transporters in the brain, especially on transporters for neurotransmitters, that is, neurotransmitter transporters or reuptake proteins. Among transporter ligands are the antidepressants, especially the selective serotonin reuptake inhibitors, SSRIs. Again, there are trademarks that may be familiar to some students, but we're not using trademarks. In addition, blockers of the dopamine transporter are used for attention deficit disorder, and others are drugs of abuse, such as cocaine and the amphetamines. We'll turn to some quantitative aspects of drugs and the brain. In week 3, we'll talk about dose-response relations, the relation between the concentration of a drug near a receptor and the response of that receptor, and this will give us a chance to define more precisely agonists and partial agonists and inverse agonists and antagonists. We'll also talk about pharmacokinetics, which is usually not discussed in detail in a course on neuropharmacology, but of course pharmacokinetics, the process by which a drug enters the brain and even enters a neuron from the outside, is vastly important. Here's an example: the Parkinson's therapy, L-DOPA, levodopa. It gets through the blood-brain barrier, and in the brain, it is converted to dopamine itself, which cannot enter the brain itself. So pharmacokinetics is a key element of drugs and the brain. We'll put all this together in a couple of mini lectures on recreational drugs. Now, the set of recreational drugs overlaps with addictive drugs and with abused drugs and with illegal drugs. Some are addictive, some are abused, some are illegal. And here is our example universe of 9 recreational drugs: morphine, tetrahydrocannabinol, or THC, ethanol, caffeine, ketamine, LSD, nicotine, again, one of our best examples, cocaine, and amphetamine. So as you can see, we're going to be covering a lot of territory. Before you go on to the next mini lecture, you may want to read my disclosures about my sources of research funding, and you may also want to take to heart the fact that I'm not a physician, that nobody associated with this course is a physician. I can't give medical advice, and you should not, on the basis of this course, give medical advice or change whatever drugs you now take on the basis of the course, or advise yourself or friends or relatives on the basis of the course. See you next time.